Welcome to chapter 38 and 39. Now in 38 I just want to go over some of the key characteristics and structures of plants. So to start it off, plants exhibit alternation of generation. Now this is kind of a different reproductive pattern where plants will typically have a gametophyte or haploid phase, which is multicellular. They'll also have a sporophyte or diploid stage that's also multicellular. Now this is different than things like animals where we have a unicellular haploid phase. That's just our gametes, sperm, ovum. And you'll see this is different from things like fungus, where they have essentially a single-celled, very basic sporophyte stage, where they have this haploid cell that goes through meiosis to get spores, but it doesn't really live as a multicellular organism or structure at that diploid part. They're almost all haploid. And so these are kind of odd. Plants are kind of odd because they have a fairly significant, in most cases, haploid phase as well as a fairly significant diploid phase. So their haploid or gametophyte phase will make the gametes. They'll go through fertilization to become the sporophyte. The sporophyte produces spores, which gives rise to gametophytes. So this process will keep continuing, but this is a fairly normal thing for plants to do, as well as some algae. So just be aware that they are kind of different, where you can have a fern plant, which if you look at a normal fern, that's the sporophyte, but underneath it you can see these leaf-looking like structures growing on the ground, which are completely separate from the fern, but those are multicellular gametophytes that are growing there that will produce the gametes, which can fertilize to give us the zygote, which becomes a sporophyte, etc. So it's just a little bit more involved than when we talk about animals or when we talk about like a fungus, where they're pretty much always just a sporophyte or pretty much always just a gametophyte or haploid in this process. For flower anatomy, because we're going to discuss things like pollination, I want you guys to be comfortable with it. So within a flower, there is the female part called the pistil, and the major characteristics of that are we have the ovary, which is what houses the ovules. The ovules are the ones that are going to contain the egg, if you will, or the ova. Okay, the female part, the female sex cells. And then you have the stigma is the top end of this pistil. That's where the pollen will attach. So the pistil is where the pollen will land, and then the pollen will grow down the stigma through the style into the ovary so that it can fertilize the ovule. You'll also see that outside of that we'll have the male part. All right, so this is going to be called the stamen, and the stamen's going to have an anther on the end of it, which is where we produce pollen. Pollen essentially is just going to be sperm, right? This is the male part. And so this will have the male gametophyte, which will be what's in each of those pollens, which are going to be trying to find, not that they think, but they're hoping, I guess, if they can hope, to land on a stigma, which will allow them to grow. Most pollen will never find a stigma, especially not of the right type of flower, and so they will never grow. Plants produce a lot of these, but very few of them will effectively fertilize anything. So that's the male part, usually just outside of that. And then as we keep going, we'll see the flowers, the petals. And the petals of a flower are really there for attractive purposes. They are there to help out pollinators generally. So they're trying to attract somebody who might be coming to interact with that flower. In some cases, they give them a place to land, but they do have a purpose other than looking pretty. They're to get the plant noticed and to get somebody else usually to come and land on it. And then beyond there, you just have their sepals, or what they call, that kind of hold everything together. But that's the structure of a flower. If somebody says something about a flower being perfect, it just means it has both female and male parts on the same flower. If they say it's imperfect, it means that the flower is just female, it just has the pistil, or it's just male, it just has the stamen. That's it. Now, once again, the significance of this is a lot of plants, especially ones that are non-flowering, will use wind to allow pollination to occur. So they have to hope that the wind takes this massive amount of pollen that they put out there and blows it to land on the stigma of another flower, ideally of another plant. Now, flowers and flowering plants have really done well evolutionarily because they've really allied themselves with pollinators, where they've said, I'll give you a reward, but in return, I don't have to produce as much pollen because you're going to get pollen on you. You're then going to fly to another flower, possibly on another plant, which is ideal, and then you're going to rub that pollen onto that other flower, ideally onto the stigma, which means we now have fertilization, so we can produce seeds and fruits. And so this is a really good system where everybody wins, they get food, but then they also make it way more efficient to pollinate yourselves, and it makes it much more likely that you'll have cross-pollination, where you're going to get pollen from one plant landing onto a separate plant. This mixes up the genetics much better. This is really what we're after with sexual reproduction. Whereas other plants are much more prone to self-fertilization, where they essentially accidentally, in some cases you could say, get pollen from one flower on the stigma of the same flower or the same plant. And so that means that you're really not stirring up the pot, you're kind of just inbreeding. And so most plants have evolved, or at least many plants have evolved, ways in which to get insects or other methods that they have within their pollen, like recognizing their own pollen and not letting it grow. They've developed ways to try to encourage cross-pollination. And obviously humans can help out with this when we can go around and specifically do it ourselves to get what we want. Now the basics of plant reproduction, the ovules, those parts that were inside the ovary, those will be what gets fertilized and become seeds. The ovary itself, that kind of base of the flower, that ovary that contains the seeds, the ovules, is what becomes the fruit. And this can be a fruit like an apple. This can be a dry fruit, as they call it, something like a helicopter. It's just whatever wraps around the seed. It doesn't have to be big and bulky and taste good. And then the seeds themselves will go through a process. If they're going to grow, if they're lucky enough that they land in good conditions, they will go through germination, which is where they do imbibition. Imbibition just means taking in a whole bunch of water to break the outer part of the seed, called the seed coat, and allow the embryo inside, that kind of baby plant, to start growing. You know, the root will come out, called the radicle, get into some water to start soaking it up, and all that water can be used to grow and elongate the cells to allow the plant to get bigger and try to find light. And so we'll call that, starting out growing of a plant germination, and it begins with this process of imbibition. Water is the critical piece. That's why typically if you want seeds to grow, you put a bunch of water there, because that's what tends to trigger it. All right. Now, the next bit we have in 39 is still going to use some of the ideas from before, but we're really going to focus on how plants can take in signals from the environment and how they can respond to them, how they can have changes occur within the plant. So this is really going to revolve around the idea of plant transport, and this is going to even revolve around eventually what we're going to spend a lot of time on, which is going to be hormones in plants. So as a quick refresher, plant transport, there's multiple ways plants can transport things. The first way is they can use these channels between cells. So there's these gaps in the cell wall where the cell membranes connect, and so we can have cytoplasm go right from one cell to another. So that's using plasmodesmata. Okay? They also would call that the symplast route. You can also have things go through the membranes, through the cell walls, called the apoplast route, but we can have things move from one cell to another, just kind of going through the actual membranes and walls. The tonoplast will be a big deal in this, just because plants also have a lot of transport that occurs into and out of this central vacuole. And the membrane of this central vacuole, that's what the tonoplast is. And so because plants do so much storage and metabolism inside that vacuole, and so they have to be very particular about what goes in and what goes out, the tonoplast is oftentimes brought up in plant cells because of that. And then we also have bulk flow. This is where plants are going to use xylem and phloem. These are kind of things like veins that plants have. Phloem tends to move sugars and food. Xylem tends to move water. And for these types of long distance flows, they're going to use pressure. And so they're normally going to have where they put a bunch of sugar into a cell where photosynthesis is occurring, and that means a bunch of water tries to go in to dilute it. And that creates pressure. And it can actually push the fluid, the sugary fluid, from cell to cell via a symplast route, via these holes, these pores, these plasmodesmata, which allows us to push sugar in a plant from one place to another, specifically from a place that's high in sugar to places that are low in sugar, like roots, or places where a lot of activity is going on, like developing a fruit. And so those are ways that plants will help move things around the cell. And at a particular level at the membrane, remember we still have all the old things going on. We have passive transport like simple diffusion, high to low concentration through the phospholipid bilayer. We have facilitated diffusion, same idea except through a protein. We're going to have osmosis we'll bring up in the next thing, but water. And then we still have active transport, where we're using ATP to concentrate things on one side of the membrane. We've talked about our good old proton pump, that's the H pluses. And so that allows us to make these gradients, these imbalances of concentration, which we can use later, because these imbalances can have power. So if we have an H plus imbalance, we get, first of all, a pH imbalance. A bunch of H pluses means acid, and then the other side's now missing most of those, so it's base. And then this H plus wants to diffuse back in. And so if we allow it, we can oftentimes have somebody else go with it, like a buddy system. So maybe by letting H plus in, we get something else done. It could be making ATP, it could be sneaking somebody else in, like a calcium that otherwise wouldn't get in. But we can use those gradients to accomplish tasks. Now, osmosis, just a quick review as well, plant cells want to be hypertonic because they want to be pressurized. That's how plant cells are rigid, is they're turgid, they're pressurized. So to be hypertonic, they need to have lots of solutes in the cell. And the more solutes you have in a cell, the lower your water potential. So if you see them show this psi, this little pitchfork looking symbol, the trident, and if you see them giving you some number with that, it's typically 0 or less, because 0 is pure water. The more stuff we put in, the lower it gets. So it becomes like negative 0.6 or negative 1.2. The lower your water potential, the more hypertonic you are. So water wants to move. So in this case, it'd want to go from 0 to negative 6. And if these were the choices, it'd want to go from negative 6 to negative 1.2. So water wants to go to the lowest water potential. So this is why for a plant cell, you want to have a low water potential. You want to have a lot of solute inside. This is why if I salt the earth once again and put a bunch of salt in the soil now has a lower water potential, it's hypertonic, and so the plant would lose water to it, the plant would die. That's why it's so dangerous to do it. So water is still going to be a critical thing for plants, but they still want to be hypertonic, low water potential. Now, as we get towards the end here of what I'll cover, quick rehash of signal transduction. So signal transduction is how we are going to receive a stimulus. So there's going to be a receptor usually on the cell membrane, although some can be inside the cell, so long as the stimulus can make it through the cell membrane. Some things that are small and nonpolar can. And so this receptor is going to be bound by the stimulus. It's going to detect it. It's going to be triggered. Usually they'll attach. And then once the receptor gets triggered, it causes a series of changes afterwards called transduction. So if you remember, there was the G protein pathway where it essentially had the G protein linked receptor, activated a G protein by binding a GTP to it, and then that guy typically activated a bunch of protein kinases, which stuck phosphate groups on a bunch of other proteins, which then did the same thing until eventually we got some type of response, which was normally activating a particular enzyme or deactivating an enzyme or activating transcription factors to turn on a gene or turn them off. And so this process can be seen within cells and within plants through a process like etiolation. So etiolation is where if you're starting to grow, like from a potato, where the plant is starting to grow, it's just starting off its life and it's looking for light. If it doesn't see light, it'll oftentimes grow white, where it's not wasting energy producing chlorophyll because it's not being hit by light. And it'll tend to grow really tall, really thin, really quickly because it's desperately searching for light. Now, once it gets light, you'll see that it'll start to send out roots more, it'll start to send out leaves more, it'll start to produce chlorophyll and turn green because now it can do photosynthesis. So this is a potato that's growing with light. This one is one that'd be like in the bottom of your cupboard where it doesn't have light, and you can see the difference between them. And that's based upon which genes are being expressed based upon stimuli. In this case, it's all about light. Now, the last slide we're going to cover for this particular podcast is just in showing hormones again, which are going to be these long distance signals. So these are going to be typically made in one part and then used elsewhere, or at least potentially used elsewhere. In plants, they tend to do a little bit less send forever away. They don't tend to send it as far away as in animals because they don't have as good of a circulatory system. Like we've got blood and a heart that makes this easier. But these are typically going to still be these longer distance signals. They're not used just by the cell that makes them or just by the cells immediately next to it. And so these will allow us to have effects where if we get some signal, it can trigger hormone production, and the hormone productions can cause these large-scale responses. Now, the example we'll give is tropisms. there's three basic tropisms that we'll discuss. There's phototropism, which is what you see below here. This is what Darwin and Went, two scientists, did a bunch of experiments on as they were doing early experiments with plants. And so this is where a plant will grow towards light. You should have experienced this where you put a plant in the window and it starts to angle towards the light. And that's going to be a response based upon hormone production. The light stimulates a bunch of hormone, which we now know is called auxin, more on that later, to grow, or I should say grow, to exist, to be produced on the dark side of the plant. And this auxin causes cell elongation. It makes the cells get bigger. And so that means the dark side of the plant starts to grow more, gets bigger, which means it pushes the plant away from it. You know, if you had your one leg, your left leg get bigger, it's going to move you towards the right. And this is essentially what happens with phototropism. So phototropism, light. There's also gravitropism, which affects different parts of the plants differently. This makes the shoot part, the stem and the leaves, grow up, and the root part grow down. And if you take a plant that's been growing and tilt it sideways, it can eventually detect this and start to adjust, where you'll get a plant that looks more like a weird sideways S. And the last type, which is my favorite, is thigmotropism, which is response to like physical pressure, like touch. And so this is why you'll see some trees that are on these windy areas tend to be shorter and scraggly, and that's because they're trying to make themselves shorter and tougher so they can deal with the presence of the wind. This can also manifest itself when you have things like vibration from music affecting plants, or crazy Russians with baseball bats, which has also been done. And so if they're going out and they are impacting the plant, whether it's through vibrations or, I mean, some people with like early plants that are growing will toughen them up by like lightly touching them, or if it's like a tree, as we said before, you can just go nuts with a baseball bat. But you can alter the way the plant grows in response to that physical contact or physical pressure that it experiences. So those will be tropisms, and that's just one of the effects that things like hormones can have. In the next podcast, we're going to talk about plant hormones, and we're going to talk about some of the other patterns that plants have, like flowering, and how those are determined and ultimately how those come about. Hope you guys have enjoyed this. See you soon.